Hello, my name is Janelle Schafer and this is a brief description of my recent journey in science. An interest in colour, art and natural food production systems preceded my current research in science. In the late 1980s I studied horticulture and this was the beginning of a fascination with botany that has continued until today. In my study of the molecules in plants, I am constantly aware of the traditional use of plants in textiles and the tradition of creating natural colours on fibre and fabric from plants. In my research, I have journeyed a little into an understanding of chromatography. The term chromatography was used by artists in the 19th century to describe colour pigments. In 1904, the botanist Michael Svet used the term chromatography to describe a separation technique which he developed to explore photosynthesis in plants. Svet developed a glass column which he filled with powdered chalk, and as the extract was poured through the column, the colours separated out into the chalk in a series of bands of a predictable pattern. In Svet's words, like light rays in a spectrum, so the different components of a mixture of pigments are dispersed in the chalk following a set pattern, and they can be determined from there. He called such a preparation a chromatogram, which means colour writing. Svet separated chlorophylls and xanthophylls using a column which he developed, now known as Svet's column. During my time as a master candidate, I have had the good fortune to receive a scholarship to attend a conference on Raman spectroscopy in biomaterials and food science in Kyoto, Japan. Our hosts treated us to cultural and scientific tours of the city of Kyoto, and we had the opportunity to explore the museum of the Shimadzu Foundation, a 170-year-old company which is a world leader in the production of chromatography equipment. In this slide you can see the first gas chromatograph developed by Shimadzu Foundation on the right, and an early liquid chromatograph on the centre left. Shimadzu Foundation has been in operation since 1875. We heard how the capital city had been moved from Kyoto to Edo, Tokyo, in 1868, and Kyoto had been thrown into poverty. The founder, Genzo Shimadzu Sr., developed an industry making physics and chemistry instruments in Japan. Early X-ray equipment, educational equipment for teaching science in schools, and later chromatographs were produced by Shimadzu Foundation. These are some early examples of the type of equipment I have used in my research. During my time in Japan, we had an in-depth look at the application of another innovative form of spectroscopy, Raman spectroscopy, which uses laser technology to explore the placement of specific molecules within a leaf. This technology is also used in medical biomaterials. We took part in an ideas contest which brainstormed different applications for this technology. These included the concept of testing food with a molecular barcode, which Professor Pizzotti has now used in his paper, Raman Molecular Fingerprints of Rice Nutritional Quality, and the concept of Raman barcode. Over the past 3 years, I have been completing a Master of Science at Southern Cross Plant Science, Southern Cross University in Lismore, New South Wales. My thesis is titled Low Volatile Extractive Compounds in Early Melaleuca Alternifolia Seedlings. For my research, I have used modern chromatography and spectroscopy methods to identify 4 acylfluoroglucanols in young tea tree seedlings. These are low volatile compounds previously not identified in tea tree. I've had the good fortune to have access to a laboratory equipped with world-class equipment and the experience and expertise of my supervisors who have nurtured my understanding of how to investigate the molecules in plants. Gas chromatography coupled with mass detectors, GCMS, has been used extensively to analyse the volatile oils in plants. The monoterpenes and sesquiterpenes are important aromatic compounds found in the Australian Myrtaceae family. In my current research, I have been analysing the less volatile compounds in one species of Australian Myrtaceae, the tea tree. Here are the 4 acylfluoroglucanols which we have isolated and identified. The acylfluoroglucanols are bioactive compounds with the characteristic hydroxy groups on the aromatic ring and a side chain carrying a carbonyl group. It has been a detailed process, first identifying the compounds and then using those pure compounds as a standard to estimate the quantity of each molecule per gram of leaf. GCMS has been a popular method of detecting the volatile compounds in plant leaves since the 1950s. These instruments are used at the Analyticals Research Laboratory at Southern Cross University to detect volatile compounds in plants, herbal products, tea tree oil, and also in food and fibre. Other researchers in our lab have used GCFID to identify fatty acids and lipids in mustard. In gas chromatography, we inject the leaf extract into a column on the gas chromatograph. Here the solute is heated to turn it into a gas and then carried on a carrier gas to a detector. In our case, we used helium gas. The columns in the gas chromatograph are very long and thin and sit in a coil in the column oven, which is heated to over 200 degrees Celsius. Gas chromatography is particularly suited to mixtures of fairly volatile liquids. The more volatile components leave the column first. Our initial experiments indicated 4 unknown peaks on GCMS, eluting later in the run, thus indicating a lower volatility, and these peaks form the basis of my research question and thesis work. We then progressed to using liquid chromatography, mass spectrometry, a system which is able to analyse less volatile components. This time, the solute is transported as a liquid. In both of these systems, the most sensitive way of identifying the compounds is using a mass detector, which can detect and record the relative masses and abundance of ions. We also use the high-pressure liquid chromatography to separate the molecules of interest. Once separated and further purified, these compounds were analysed using nuclear magnetic resonance spectroscopy, NMR. This system uses a very large magnet to probe the intrinsic spin properties of atomic nuclei. NMR is used for the structural determination of small molecules and is used to analyse mixtures in food chemistry, biochemistry and physiology. Finally, using LCMS, we measured the quantity of these pure compounds in a specific weight of fresh leaf material, concluding the low volatile acylfluoroglucanols measured around 10% by leaf weight of the more volatile terpenes. Currently, we are looking for the signals of potentially larger low volatile molecules in the young tea tree leaves. We are using the LC-QTOF. This machine uses liquid chromatography, time of flight analysis, and is very sensitive and can pick up many more signals than the LCMS and GCMS. However, the accuracy for detection of an exact compound is limited. It will instead point to further research possibilities of low volatile compounds in young tea trees. Using this technology, we hope to provide a dictionary of active molecular weights in the tea tree plant that may lead to further research that will provide molecules of use to the agricultural industry. Tea tree is a plant with a diverse range of biological applications, useful in medicinal applications. The possibility is there that these modern instruments and technologies can assist us in understanding further roles for tea tree in producing compounds that can safely be used in agricultural systems.